Mike Caligiuri, and I'll be the moderator of this session, Challenges in Trial Design for Immunotherapies and Combinations. And what I'll do is I'll just, for the sake of time and staying on time, we'll have our speakers come up at the end, and we'll do questions at the end. And I'll just announce our speakers now. Their bios are in the book for those of you that want to look at the bios, and I encourage you to. So our first speaker will be Rich Simon from the National Cancer Institute, who will speak about novel trial designs to assess early clinical efficacies of immunotherapies. And then Rich will be followed by Lisa Butterfield from the University of Pittsburgh, then Harpreet Singh from Immatics Biotechnologies, followed by David Porter from the University of Pennsylvania. And each of those will have, I think, 20 minutes, and then our FDA perspectives will be 10 minutes each, from Raj Sridhara from the FDA's Center for Drug Evaluation and Research, and Peter Bross from Center for Biologics Evaluation and Research. So we'll start with Rich Simon, who'll talk about novel trial designs, and then I'll ask our other speakers to come up as Rich concludes his 20-minute presentation. Thank you.
Thanks, Rich. Thank you very much, Mike, and thanks to the program committee for inviting me. I will talk about trial designs, some novel, some not so novel. Immunotherapy, and everyone see that, is different, and I think it's different in several ways. One, it's different because it actually works for patients with metastatic disease, at least for some patients with metastatic disease, producing durable, in many cases, complete responses that have never been seen before. And so I think it necessarily grabs our attention to see how can we extend and improve even those results. But it basically is the hottest and most exciting therapy available today. And it's also different for various other reasons, important reasons that have been mentioned already this morning. Limitation of adequacy of preclinical models, more complex mechanism of action. You're talking about the immune system as well as the tumor, inapplicability of traditional phase 1 designs, which can be very important because of the necessity of combination regimens. So here, you're necessarily talking about regimens that have multiple parts. You need to be thinking about regulatory T cells as well as directly what you're doing to the maybe the CD8 population, and so it's likely that to really improve the regimens, we'll wind up with regimens that have even more components than what we're seeing today, and also numerous candidate immunomodulating agents that need to be evaluated in combinations with current regimens. And I think these are not details; these are profound differences that influence not only clinical trial design, but the whole culture of immunotherapy development. And I think, as was described this morning, we need to be cognizant of that, because it really may, if we really want to take advantage of what we see in immunotherapy today and moving that forward, we're going to have to maybe change some paradigms. There may be a bigger role for public funding of some aspects of immunotherapy design, because as was mentioned this morning, some kinds of immunotherapies may not be, there might not be a great business model for the pharmaceutical companies, and even in cases where there is, public funding will probably have to carry things and move them along further than in a much simpler situation of small molecules. We've talked about some of the issues of phase 1 issues and preclinical pharmacology evaluation. I just wanted to mention I collaborated with Samir on a study in which it involved a literature review of a large number of vaccine clinical trials, basically showing that the incidence of grade 3 and 4 toxicity in the use of vaccines is exceedingly low, generally less than 1% of patients or less than 0.1% of vaccine administrations. And we wound up, in the cases where there is dose grade 3 or 4 toxicity, there's really very little evidence that it's toxicity that is dose related. And so we wound up proposing another development design for that, in which basically you start off with a biologically active dose of the vaccine and its adjuvant, and then you combine that with the immunomodulators that you're going to use, and any kind of dose modification that you have to explore. You basically explore using the vaccine in combination, not as a single agent. Finding some optimal dose as a single agent isn't really going to get you where you want to get, because you're going to have to be using some immunomodulators with it. I wanted to mention one of the paradigm kind of issues with discovery rather than hypothesis testing. Sort of we're used to doing clinical trials and finding out, testing hypotheses about the contribution of some drug in the context of maybe a particular histology of disease or particular genomic alteration. And we design a trial with a type 1 and type 2 error, and the focus is on that particular trial. And the type 1 and type 2 errors we're sort of familiar with, the chance of getting a false positive or a false negative conclusion from that trial. And when we're in more of a discovery mode, which I think we have to be in developing even more effective immunotherapies, we have so many candidate modulators we can use, and in the ways we use them, we are more in a discovery mode, and we have to think, I think, somewhat of the what I've called the type 3 error. And when we do measure clinical endpoint, but we also have the opportunity, I think, to do intensive immune surveillance with things like single-cell RNA-seq is one example. And what is a type 3 error? The type 3 error is the error you make by not studying the intervention. What you could have discovered had you done a study, even a small study, that included that intervention. Large treatment effects are observable with small sample sizes if you do the study. And so when you're in more of a mode, a screening mode, or we don't think of it as screening, except screening for large effects, either clinical, preferably clinical, but we also have other intermediate endpoints that can be measured in an immunotherapy trial. We need to bear in mind, at least, that there's an opportunity cost from doing a large trial on one combination rather than smaller using the same population or the same batch of money to do more trials on a variety of combinations. There's obviously a need for one of the potential roadblocks is there's a need for companies to make immune modulating agents available to academics for these discovery studies, particularly with novel combinations. And some right now, getting available some drug can be a very long, drawn-out process, and I think that process and the difficulty of having different immune modulating agents from different companies is one of the serious roadblocks that somehow needs to be tackled if we're really going to make maximum progress. Now, I was going to go over just a few phase 2 designs and then mention one phase 3 design. I think obviously, and these are not particularly novel, but one obvious one is basically for example, you could take a patient who would be a good candidate, say, for an anti-PD-1 checkpoint inhibitor, and if the patient doesn't respond to that, you could treat the patient with that anti-PD-1 plus some immune modulator to see to see whether you, and then if you get responses in that context, that's good. And you can use that same sort of setting if you can actually arrange to do a clinical trial randomized among several novel combinations, an anti-PD-1 plus X or an anti-PD-1 plus Y or an anti-PD-1 plus Z. There are some advantages and some disadvantages to trying to do that in one trial. Certainly, if you're using one assay for evaluating all three of these, then things are going to be more comparable than if you're doing separate trials with separate assays. Of course, it's harder to set up this kind of a trial than it is a trial of one novel combination. And another thing that may have more applicability for immunotherapy than it has had in the past for chemotherapy is the idea of factorial designs, where you have an X and a Y immunomodulators that you can potentially add to an anti-PD-1 antibody, and you have 4 arms, anti-PD-1 alone, or anti-PD-1 plus X, or plus Y, or plus X and Y, but you analyze it essentially as a two-drug, and you size it as for a two-group comparison rather than for a 4-group comparison. For example, if you want to evaluate X in its contribution to the anti-PD-1 antibody, you compare the 2 arms containing X, some of which contain Y and some of which don't, to the 2 arms that don't contain X, some of which are PD-1 alone, anti-PD-1 alone, and some anti-PD-1 with Y. That's the idea of the factorial design. Very often, it's not feasible when you're dealing with chemotherapy because the dose of X precludes delivering the full dose of Y and vice versa. But if you're dealing with components here which are not highly toxic, don't themselves cause dose-limiting toxicity in this context, then you have that opportunity to use this design a little bit more. I would say phase 2 design for discovery, I would distinguish from a phase 2 design really driven by regulatory considerations. So obviously, there are some regulatory considerations that deal with safety that are important for any kind of a phase 2 design. But for others, we can potentially think of doing small screening designs that may not be appropriate in a regulatory context, but would tell us what we want to study more fully. And so I think we need to think broadly about the options available to us. And I really I'm not going to spend too much time on endpoints. We have the particular issue with immunotherapy that although some of the responses occur very quickly, other responses can be very delayed, and even after a traditional progression of disease. And so that creates some issues, which I think may be discussed later in more detail. But you can see this in some of the I think this was for ipilimumab, the top being a survival curves and the bottom being progression, the time to progression curves. And you can see, looks like a lot of patients had very rapid progression, but possibly they may have had still some survival benefit. So I think in these kind of studies, we have to think in this term and perhaps define the duration of therapy independently of what progression of disease is looking like, at least for the arm containing the immunotherapy. I think this is the same kind of thing for an anti-PD-1 antibody. I did want to mention also briefly, I'm sorry, this cover page doesn't show up, but it's a design that I published with a collaborator, Funking Hong from Dana-Farber, a run-in design for which I think may have applications in phase 3 trials. So the idea is, one application of it would be you'd screen patients using, say, broad eligibility. This is for a situation where you don't really know what's the best predictive biomarker, but you potentially want to gain the efficiency that you can sometimes get from enrichment designs where you select patients for those who are most likely to benefit. So you do a run-in on the test treatment. So you're going to compare a test to a control regimen. And before you do the randomization, you do a short run-in on the test treatment. And then you, in this case, evaluate some intermediate endpoint, like it might be T cell, whether the T, how many T cells are activated, or interferon gamma level, or whether the T cells are expanding. You do that, and then you basically, in this case, you would have the patients who are not showing that immunological response go off study. The patients who are going in, who are showing the response after this short run-in period, then get randomized. And so you have used essentially a post-treatment marker as a predictive biomarker. Now, this, of course, is for the situation where you think you know what the right predictive biomarker is, but it turns out it's something you can only measure after a run-in on the treatment. The other version of this would be where you really don't know, and so you would, whether you randomize somebody or not would be independent of measuring these short-term candidate predictive biomarkers. But after a short run-in period, you'd measure a bunch of those kinds of things. And then at the end of the trial, you would be able to, since you've randomized everybody, regardless of that information, you'd be able to sort of do an analysis of subsets based on positive for any of those candidate biomarkers. And it turns out it can give you very good increases in power and reductions in sample size. Finally, I have a couple of slides. I want to say, I think we need increased public support for a couple of things. One, experimental and computational initiatives for identifying new antigenic targets for adoptive T-cell therapy. I think that was alluded to this morning by Dr. Fox. We know about the expression of lots of receptors in a tumor, and all of those receptors are candidates, at least, for immunotherapy, depending upon what other kinds of cells they're expressed on, and whether they're likely to be presented on MHC molecules. But I think there's a lot that can be done with a very well integrated experimental computational initiative for identifying new antigenic targets. The CAR T-cell story is terrific for CD19, but once you get beyond CD19, so far it's not that exciting. So I think the issue is, well, how can we extend that? And I think it's important enough that we can't just sit back passively and hope that problem solves itself. And secondly, I think we need an integrated experimental computational systems biology modeling approach of the T cell effectors and their relationship to tumor and immune system. We're beset with more more candidates and more candidate combinations than we really can test just empirically, and I think we need something to go along with all of these empirical studies, that we use data from those studies to put it together to help us be building a model as we go along that that helps us think through issues of timing and issues of how this very complicated system works together, and I think there's value in that approach. Thank you.